All right, so pancreas secretion regulation. Three basic stimuli, okay? One, parasympathetic nervous system, so acetylcholine from the vagus nerve. Two, cholecystokinin, all right? Cholecystokinin released from the duodenum in response when there's chyme present, okay? So when, in other words, when digestion is happening or is initiating. And then secretin, secreted by the duodenum in response to acid, so low pH. And that secretin travels almost immediately to the pancreas and increases bicarbonate secretion, right? So we have a nice, tight little control system. So the first two, parasympathetic and cholecystokinin, promote enzyme formation, and then secretin stimulates bicarbonate secretion, right? Because it's the acid producer side. These all work better together, right? So like you get your maximum secretion if you have sympathetic, cholecystokinin, and secretin all working together, which is the typical case, right? You know, usually you're going to have all three of those happening. Secretion by both the stomach and the pancreas falls into three categories, or three phases: cephalic, gastric, and intestinal. All right, so this is on the pancreatic slide, but this also applies to stomach secretion too. Okay, so the cephalic phase comes from the brain. It's CNS driven. You know, so you know how like your stomach starts to growl and you maybe get a little drooly ahead of the time that you're going to eat, right? That's all from the brain. So when thoughts of food and anticipating food stimulates the production of enzymes so that the body's ready to digest. But we don't get a lot of the bicarbonate and water solution until food actually enters, okay? The bicarbonate water solution can be made really fast and easy. The enzymes, like all enzymes, are proteins. They take time, right? So the idea here is, behind the cephalic phase, is by the time food gets to the intestine, we want digestion ready to happen, right? So the only way that's going to occur is if we prep ahead of time. Pre-game, as we like to say, right? The gastric phase, now food's entering the stomach, and the stomach senses food in the stomach, and it responds by stimulating itself as well as these other materials. So it's kind of a feedback loop. Remember, gastrin comes from the stomach and stimulates the stomach in response to food being present. Then the third phase is the intestinal phase. Now, this is also sometimes called stomach emptying or gastric emptying, right? So here, it's the chyme has started to enter the duodenum, right? Now, the duodenum is going to be affected by the characteristic of that chyme and is going to trigger hormone release to tweak what the stomach is doing to best match, right? So when chyme starts to enter, we get secretin production. Now our bicarbonate solution is being produced. The enzymes, which were made from the cephalic phase, once secretin comes on board, they get washed into the intestine, right? So those enzymes start to move and enter the lumen where they can do their job, right? And those two things together then initiate digestion. So cephalic, gastric, and intestinal are these three phases of sort of gastric secretion. The intestinal phase will continue until the stomach is empty, right? And then it's sort of paused, you know, the stomach is at rest until the whole cycle happens again, right? Okay, so you've heard me talk a lot about the duodenum, and I've told you before that it's kind of the control center for the digestion and secretion aspects of the gut, all right? So let's do some details. S cells are present in the duodenum and the proximal jejunum. They produce secretin. The trigger is a low pH, right? And the response, so the secretin is absorbed, goes to the pancreas, triggers additional bicarbonate formation. Why? Here's our neutralization reaction, right? If we take stomach acid, HCl, We add sodium bicarb, because that's what we made. We got plenty of sodium because we're humans. Bicarbonate, because we made that. That's going to flip into NaCl and carbonic acid, and then those break down. So you'll notice that our hydrogen ion has effectively been removed, right? So we've neutralized the acid. So what comes out of the stomach is usually 3 to 4 pH, right? The bicarbonate solution takes it up to 7.5, 7 to 8, okay? And that's where our pancreatic enzymes work best. The other reason this is done is acid is very hard on the mucosa of the GI tract. It's only the stomach that has these optimizations for protection. Now, the whole intestine has a mucus coat, but it also has a lot of folds and ridges, which makes it hard to protect. So we got to get rid of the acid early on, and that's what this secreting system does. All right, so that's S cells and the neutralization. CCK, cholecystokinin, comes from I cells in the duodenum, and here it's in response to products of protein digestion, as well as presence of fats. Both are triggers of CCK. CCK's big role, there's two I really want you to remember. CCK is the gallbladder hormone, okay, so it triggers gallbladder contraction. But CCK also, it's the most potent stimulator of pancreatic enzyme secretion, right? So CCK is released in response to a heavy meal, so to speak, and it's the most potent stimulator of all of these actions. All right, so given the fact that the duodenum is detecting some of these different changes and responding accordingly, we can sort of see that in process, right? So if we put hydrochloric acid in the duodenum, we get a big burst of water and bicarbonate production. If we take peptones, which are protein digestion products, and put those in the duodenum, we get a lot of enzymes and a lot less water and salt. And then the middle one this is kind of a combination of both, where we have both hydrogen ion high acid, but we also have protein digestion and some fat digestion too. So fats, I know it says soap and that's what it is, but basically if you metabolize fats, you get soap. Okay, so soap is seen as a fat digestion product. So the body responds with both things, both bicarbonate and enzyme production. All right.